In progress.
First, I know that there were some issues with the exam not opening up, but I know that at least one person has taken it at this point. I tried yesterday with McGraw-Hill being closed yesterday. I tried just to reassign it and see if it would, whatever had happened would be fixed. And someone has taken it. So those problems should be fixed. If you're still struggling with it, call McGraw-Hill. They are open Monday through Friday from, I think, 8 to 8. I think they're also open on Saturdays. Are there other questions or issues?
Oh, is everyone else having trouble accessing the exam as well?
There was one other person. Ashley, when you send me emails, they come back to me. They, I don't know why they're coming back to me, but when I send you a response, it comes back to me. Do you get any of my responses?
Yeah, I get your responses. I send it both. So I send an email to both your internal and external account.
Okay.
Because I don't know which one is, because I don't know like which one you get faster.
I think I get both of them, I think.
Oh, okay.
Yeah, I was just making sure because I thought, why is it bouncing back to me? And so as long as you're getting it, yeah, so that's when I reassigned it again yesterday, and at least someone has taken it. And so see if you can do it now.
Okay, let me look.
Are there other questions or issues?
I was actually wondering, it says on the syllabus that the case study should have opened up or something, or should be opening
I
up.
wasn't gonna have it open up until we did bone.
Okay.
Because I wanted
I was
to
gonna say
actually
it
be
didn't open for
something.
me.
Yeah, it's not there because we haven't done bone. And I want it to be actually be something relevant. And it's gonna be over bone.
It looks like it let me into the exam. I'm gonna exit out before I start it.
What?
It let me into the exam.
Okay,
I just
good.
said I haven't started it.
Yeah, I don't know what the problem was. It just may have been that there was a fluke, but it works now. Go ahead. Other issues?
Professor Gessel,
Yeah.
I was just wondering if you could clarify which Wiley lab exercises are due tomorrow, because I know for this section there was a couple.
8 and 9.
Okay, thank you.
Yeah, if you look on the syllabus, the due dates are there, but it's 8 and 9, and we will go over 10 on Wednesday. Other questions? Oh, you know what? Oh, that's not what I wanted. There it is. So we will finish the cells tonight and possibly start chapter 7 on bone. So, all right, so I think where we ended was talking about this resting membrane potential. Why do they call it that? Why would something be called a membrane potential? It's because there's a separation in charge across the membrane that is a form of potential energy that can be used for later purposes. So if you remember the difference between a potential energy source and a kinetic energy source, a potential energy source allows you to use it later. It allows you to store it with the intent to use it later. So you separate this charge and then you let it flow. Remember how at the very end of last week, I said, think of it like a battery. You have a battery at home. What the point of it is to plug it into something, create a circuit, and let that charge flow. And the battery will die when there's no longer you can no longer have a separation in charge. It's the same thing here. You're creating this separation in charge across the membrane with the intent that you can let it flow later and let it work for you. So the only difference between this and your battery at home is this: most of your batteries are not rechargeable. This is a rechargeable battery. Where you keep resetting the resting membrane potential back to where you wanted it. That's the only major difference. So all cells exhibit this resting membrane potential, ranging from a -50 to a -100 millivolts. Still think of it like your battery at home. Your typical battery, 9-volt battery, meaning there's a separation in charge of 9 volts. So the inside of the membrane is -50 to -100 millivolts more negative than the outside of the battery. One of the major reasons for this is the sodium-potassium pump. What can somebody tell me about the sodium-potassium pump? I know we talked about it. And we talked about it in the fact that it was a certain form of transport and it worked via the transport.
So like
How does it work?
well, when it opens, the sodium, you know, cross the threshold, but then in it where the potassium from the other side, it's almost like they kind of swap places, sort of.
Right, right, right. And so what you're right, Christina, good job. The reason they're swapping places is because you're building up a higher concentration of sodium outside of the cell and a higher concentration of potassium inside the cell. And you're pushing these things by 2 potassium going into the cell, 3 sodium going out. And you do it by hydrolyzing ATP. So this is active transport. Cells that have this resting membrane potential are considered to be polarized, meaning they have 2 separate poles, just like your polarized molecules that have a separation in charge across the molecule. Have a partial positive charge, partial negative charge. Same thing here. Are there questions about this? So how do you maintain this resting membrane potential, or how do you keep regenerating it or recharging it like you would your rechargeable battery? It's mainly maintained by the potassium concentration gradient. Remember, there's more potassium inside than outside. And in addition to the potassium ions, there's also a lot of negatively charged proteins in here. And between the sum of the positive charged potassium and the lots and lots of negatively charged proteins, the result is that the inside of the membrane is a -50 to -100 millivolts, less than the outside. Also, the membrane is somewhat permeable to potassium due to leakage channels. The ions will diffuse according to their electrochemical gradients. Remember, I think we talked about this before, electrochemical gradients. Chemical because it's a concentration gradient. Electrical because it's got a charge. So essentially you're looking at the concentration gradient of ions. That's an electrochemical gradient. So the membrane is somewhat permeable to potassium due to leakage channels. And the sodium potassium pump, in addition to the other thing that helps maintain this, is the sodium potassium pump is going to pump 3 sodium into the extracellular fluid and 2 potassium into the intracellular fluid for every ATP hydrolyzed. And so the extracellular fluid is going to contain much more sodium than the inside. And it's also, the charge is also going to be balanced. The sodium is going to be balanced, just like the negatively charged anions are doing for potassium inside the cell. The sodium is being balanced by chlorine on the outside of the cell.
I have a question.
Go.
So when it comes to sodium, I know we're supposed to have it in our system,
Yes.
but is there a thing as too much sodium? Because I know, like,
I'm sure there is. I think because it affects blood pressure. I don't know where that limit is, but if you become hyponatremic, you're gonna die, which means that you run too low on sodium. I think you need to have a balance of it. And I don't know if it's so much the sodium that's giving, well, yeah, it is. Never mind. The sodium is what essentially is also regulating your blood volume. And that's why high sodium is related to blood pressure. But if there's a balance with how much you should have. I would be really surprised if, it's one of those trace metals or one of those ions you have to have. So there's a balance with how much you have to have. Not having it at all is not a good idea either. We like it to be low, but we don't like it to be absent. Does that make sense?
Yep.
When next semester, when you study kidney function, you'll learn all about how water follows sodium into helping maintain your blood volume.
I have a rough idea of that because I work in dialysis. So like I kind of have an idea of what
Yeah.
sodium does to the body.
Yeah.
But like I know like if we get if there's too much, it can be detrimental. So I was just wondering if that was correct in this type of context as well.
I'm sure this is where I do. In this case, I don't think if you are hypernatremic, meaning you've got too much sodium, you can guess where that natremia is coming from. If you look Na sodium, bear with me. As I'm using this word, I'm gonna change screens for a quick second.
It's actually a really short range. It's only a 10 milliequivalent per liter range that you're supposed to be in with your sodium levels before you go
Really?
hypo or hyper.
Really?
Yeah, between 135 and 145 milliequivalents per liter. Hyponatremia is anything less than when your blood sodium falls below 135.
Yeah. And what tends to happen is, just to add to what Justin definitely knows more about the numbers than I do there. What tends to happen is people who are put on diuretics, which you've probably seen in dialysis, people who are put on diuretics can become hyponatremic. They can also become hypokalemic. And the result is that they lose the membrane excitability.
And I know they give them KX-late to that, to like
What is
reverse.
it?
They give them KX-late, it's called. It's like a small drink. If somebody has like, high potassium level,
Okay.
especially patients that go to dialysis, they give them KX-late so that it reverses that and like neutralizes it. So it's like within the number that they're supposed to be.
Huh.
Yeah, we have like designated sodium baths and everything, like if depending on what's going on within the patient, but the doctor or the nurse practitioner usually tells us what to give the patient. But I was just curious in regards to like within this scope, within like this class, like if it was the same thing.
Yeah, it is the same thing. I mean, I'll tell you, and you guys, I share graphic things with you. I share emotional things with you, partly because I know you'll remember it. Like me telling you how they cervically dislocate mice. I can tell you that I am, if you've noticed all my animals around me, I'm a big animal person. And having watched that once or twice, I'll never forget watching it because I don't like doing animal research. I don't enjoy it. And most 99 of people that do animal research don't enjoy it. But I'm really glad I don't do it now. I worked with chick embryos for a long time, and that was the best I could do. because they were young enough that they didn't even realize I was there. And they didn't even realize something was going on with them because they were only you'd consider a few weeks into gestation. But so I share these things with you that are graphic, sometimes a little unnerving because you'll remember it. But so what I'll tell you is the way that lethal injections work is to give a very high dose of potassium chloride. And the result, and you can see what the result would be just from looking at what we're looking at, the membrane potential. Can you see what the result would be?
Yes.
Yeah. Is you eliminate the membrane potential. And so there's a certain amount of, especially sodium, because sodium is what allows water to flow. So, there's hormones, aldosterone, predominant hormone that's involved in maintaining your blood volume. And what it does is it increases the uptake of sodium in your kidneys. And it increases the uptake of sodium knowing that water will follow. So that's just one of those examples of why sodium, we don't want too much of it, but we, like Justin said, there's a very little balance. But many times your kidneys will keep your sodium in check because they will get rid of sodium when it's too high, if they're functioning normally and not under the influence of other stuff. They'll do it all on their own because that's their job. But I think when it gets excessive, then you reach a threshold, just like if you're if there's protein in your urine, there's only so much your body can do to take in what and return and reabsorb what you need to in your kidneys. And when you go beyond that, then you end up with protein in your urine, which you should never have on a normal person. So normally your kidneys take care of that if they're functioning right. And if you're not going excessively.
Does that happen as a result of like the membrane being weak, like ketoacidosis?
It possibly, I don't know exactly when it comes to ketoacidosis. That sounds like something else because that's usually you're burning too many ketones. And that's a metabolic disorder.
Gotcha.
If you'll let me study up on that one, because I have to study up on the urinary system because I go over in 122 and in my physiology class at Stevenson. I'll study up on that and I'll tell you. But I don't think, I think that's related to the kidneys a little bit, but I think there's more to it.
Got you. I thought it was protein leaking.
Is that what ketoacidosis? I mean, from what I understand, it gives you a fruity smell to your breath also.
Right. Yes.
And if my mother was home, she would be great for that question because she was a diabetes nurse practitioner. Let me study up on it a little bit and I'll ask her when she gets home too.
Thank you.
You're welcome. Most often protein in your urine can come from membrane damage to the kidney membrane, just kind of like Zena was just saying. And I wonder.
I work in nephrology 2 days a week, and I think sometimes it can actually be genetic, kidney like AKIs or kidney disease, or even proteinuria can actually be considered a genetic disorder too,
Right,
or it's like a pre-existing like risk factor or something.
right. Yeah. I think there's a lot of different factors. I know that, you can have creatinine, which is a protein breakdown product in your urine, especially if you're working out a lot. But having an excessive amount of creatinine in your urine is a bad thing. You can destroy your kidneys with too high of protein breakdown. There's a disease called, oh shoot, I knew what it was until a second ago. Myelitis, that will actually destroy your kidneys because it ruins the membrane.
Nephritis.
No, nope. It actually it is muscle breakdown. Bear with me here. I worked for a long time for Walgreens. I actually only quit there about a year and a half ago. But while I was there, one of my managers came up to me, rhabdomyolysis. The major issue that comes with it is you get kidney function dysfunction and you destroy your kidneys. But he came up to me one night at work and he said, it was just he and I at that point in the night. And you may not be seeing my screen, it says it's paused. He came up to me at the end of the night and he just made the mention to me. He said, I think I'm going to go into the ER after work. And I kind of looked at him and I was like, why are you going to do that? And he had it in the past and he could tell that he was dealing with the symptoms of it then. And he ended up going into the hospital and being there for another week and a half. And my mom has some muscle degeneration on her hand here. And when she showed it to me, I got all concerned. I don't know, she's seen a hand specialist and that might be what caused it because they were doing some electrical conduction studies. But what this does is it essentially just clogs the membrane of your kidneys. And it is protein breakdown.
We have a patient in nephrology that has calciphylaxis.
Say it one more time, I'm sorry.
I actually have a patient that I've seen today. She has calciphylaxis and it's affecting her kidneys.
Okay.
And it said something along the lines of that that it's starting to cause her to, it's blocking some of her kidneys and her kidney functions because the calcifications that keep occurring in her body.
Right. Yeah, that would make sense. Yeah, that membrane is really important and all your blood pumps through it within a matter of, I think, a minute. It's very similar to your heart in its pumping function. Not so much pumping, but filtering function. I think if I remember right, and I'll bring it up in a couple of weeks because I'll go through it probably in my physiology class at Stevenson before I go through it in here. But you filter all your blood within a matter of a minute, and you will filter all of your blood about 5,000 times a day. A sec. All right, then we got to get back on track because we're getting behind. Give me 1 second. I'll let you know because I'm going to look it up in my book that I've got here on my table. This is a physiology book that I use for Stevenson University. So give me 1 second here. I think I got the numbers from this book. Maybe I'm not going to find it fast enough. Oh, wait, maybe. Okay, I'm not going to find it fast enough. But I know it's several hundred times a day, if not higher. All right, did we answer your question about sodium? That's where that started.
Yes.
Okay. This is a great class for getting off on tangents. Some of them I instigate, some of them you instigate, and I should be better at getting us back on track. But I enjoy the fact that we have as much conversation as we do. Because you'll find that a lot of online classes have little to no discussion sometimes. So the way that you activate neurons and muscle cells is to upset this resting membrane potential. So you allow that current to flow, just like you would your battery. And then you use it. So just like allowing an electrical wire to run, you do the same thing with regards to neurons and muscle. So cell environment interactions. This is another thing that has to do with the plasma membrane. So we talked about the resting membrane potential. We talked about, at the very beginning, we talked about different integral proteins. Now we're going to talk specifically about cell adhesion molecules, which was one of those integral proteins. So they can function as molecular Velcro that's going to anchor cells to the extracellular matrix space and other cells. They can also be arms that are used by migrating cells to haul themselves past other cells, allowing them to move by other cells and slip through blood vessels and capillaries specifically. They can be SOS signals on the blood vessel linings that signal white blood cells to nearby infected or injured places. If you remember, Zena, we talked about integrins when we were talking about in the immune system in biology 122. That's what they're referring to here. Mechanical sensors that respond to local tension at the cell surface by stimulating the synthesis or degradation of adhesive membrane junctions. And they can work as transmitters that can direct cell migration, proliferation, and specialization. So how do they work like these signaling transmitters? Through receptors. So these membrane proteins, CAMs, that can work as receptors and specifically bind certain ligands or ligands, hormones, neurotransmitters, paracrines, some other cytokines and so forth. Contact signaling is important in cell recognition to allow for necessary development and immunity. When we talk about the immune system, we talk about cells that come right next to other cells. So they're right up against their face. And look at them. And if there's something wrong with that cell, they kill it. But that's their job. We have cells that make cell-to-cell contact with your B cells that make your antibodies, and T cells that activate them to create your immunological memory, which is why you get good vaccines. You get good reactions to vaccines and you develop a secondary memory. We also have chemical signaling by binding specific ligands. Different cells can respond differently to the same ligand based on what receptor they're using. We have different receptors for the same ligand in many cases that will result in a different reaction depending on what receptor binds the ligand. These receptors that bind these ligands can be catalytic enzymes, chemically gated channel link receptors, or let me back up. Chemically gated meaning that the ligand binds and it opens or closes the channel. Or they can be G-linked receptors that bind to second messengers, such as cyclic AMP. Remember, this was one of the derivatives of adenosine, adenosine diphosphate that's part of RNA. So cyclic AMP is a second messenger, and it stimulates, it gets created so that it can work as a messenger and transduce a signal into the cell and amplify a signal into the cell. So a ligand binds to a receptor. It activates a G protein. This G protein has what's called GTP attached to it, and that's why we call it a G protein. So like ATP, adenosine triphosphate, we have an guanosine triphosphate that's attached to this G protein. So the G protein gets activated. It comes over and it activates cyclic adenylase. Cyclic adenylase is going to create cyclic AMP. And cyclic AMP then goes on to activate a bunch of other things, including kinases. There are enzymes that are involved in phosphorylation of multiple things. Phosphorylation, I think we did talk about this. Phosphorylation is a really good switch. Turns things on, it turns things off. So you can put a phosphate on something to activate it, and you can take it off to inactivate it. We talked about it when we talked about, bear with me here. Example of this right here. ATP gets hydrolyzed and it attaches a phosphate onto this carrier, the ATP, excuse me, sodium potassium ATPase pump. This is a phosphorylation. So kinases are enzymes that specifically phosphorylate other things. That you can add a phosphate and activate things, or you can take away a phosphate, or excuse me, you can add a phosphate to turn things on, turn enzymes on, or you can remove a phosphate to turn them on. Oh, so either way, you can inactivate or activate an enzyme by either adding a phosphate or removing one. Questions?
When you mentioned that this process helps with like the body processing the vaccines, could we develop an allergic reaction from this, or would that be from one of the other ingredients? Like, or would that be like the physical ingredients?
It's not from this that you're going to develop an allergic reaction. I will tell you that you almost always develop some sort of allergic reaction to, you're going to develop, what is a vaccine?
It's usually the dead virus, isn't it?
Well, sometimes it's not, or it's a really tiny part. It's not always a dead virus. It's a fragment of a virus or a fragment of a substance. It's some sort of antigen. But I can tell you that an allergic reaction is very similar to an immune reaction. And you have to have an immune reaction to a vaccination, because that's what it is. I know that's a really simple explanation. But you're going to develop an allergic reaction. I know when I got my second booster to the COVID vaccine, I developed a major bump on my arm. And you're going to develop a reaction, and you should, because it's just like you developing any other immune response. Usually people who struggle with allergic reactions, that's why, is because they've been exposed to it previously. But 99% of the time, those allergic reactions are not going to be bad. I'm learning a lot of things when it comes to, I don't know if I shared it in here. I recently read a book about the history of autism and how people got very scared of immunizing their children as we watched the rates of autism grow. And the study that was done on that was crap. He had no controls, and sometimes falsified his data. And what I think when it comes to, I'm all for vaccinations. I would love to see everybody get vaccinated for COVID, because there are people who cannot be vaccinated. And do people ever die of vaccines? Yes. I think people, having not been a parent, it's really easy for me to say this, but if you get a child vaccinated, you should be watching that child for at least the next 48 hours and watching them carefully. because when I hear stories of children getting hurt from vaccinations and dying, usually it's over a day. I can't imagine that you couldn't see that your child was going downhill. And I'm not a parent, so I have to be careful in how I say that. But I can't imagine that you wouldn't notice that they'd become very lethargic. I mean, if they're getting really upset and running a really high temp, why aren't you taking them into a doctor again? And so I think there are people who respond to vaccines. I have a nephew who is immunocompromised. Genetically, he has a deficiency. And he almost died at 3 months after being vaccinated. And he will never be able to be vaccinated again. But I think people should get vaccinated to protect those who cannot be vaccinated. There's a herd mentality, or you can develop a herd immunity where you need at least 85% of the population to be vaccinated. But all right, I'm going to stop. Yes, you can develop an allergic reaction because I think you're supposed to, but can allergic reactions become deadly? Yes. Does that help, Lauren?
Yes.
Okay, and I'll shut up now and I'll get off my soapbox about immunizations.
We can talk about it later after class.
Okay. Yeah, and if you disagree with me, feel free to talk with me about it later after class, because I like the input. My brother and I, he's a lawyer, and he and I can get going because he's kind of anti the vaccine. He finally got it recently, but they have real issues with it where I don't. So it's kind of interesting. And we still get along and we still talk. So voltage sensitive membrane channel proteins that function in electrical signaling. They respond to changes in the membrane potential by opening or closing channels. And Lauren, I actually really liked your question. And the reason is because I never really thought of it as an allergic response. But that's exactly what it is when you develop an immunity to it. You have to have some sort of allergic response because normally it's something that's non-harmful to you. And then you develop an immunity to it. And then when you get hit by something that's more harmful, that's like that, like the real virus, then you're now protected or more protected than you were before. All right, now I'll shut up about that. Okay. So voltage sensitive membrane channel proteins that function in electrical signaling. They respond to changes in the membrane potential. So these are, rather than having a ligand bind to a channel and open the channel, you're having a voltage change open the channel. We will spend a lot of time, once we get through bone, talking about bone, we will start talking about the rest of the semester spent on the nervous system and muscle. And a lot of that is based on voltage changes and voltage gated channels. So cytoplasm is the cellular material between the plasma membrane and the nucleus. Cyto meaning cell, plasm meaning like a slimy gel-like substance. Cytosol is a viscous transparent fluid in which other cytoplasmic elements are suspended. So the cytosol contains the cytoplasm, and the cytoplasm is a cellular material with all the organelles. The cytoplasmic organelles are the metabolic machinery of the cell. where each type of organelle carries out specific functions. One example is the nucleus. Inclusions are things that are not organelles. So anything that's in the cell that probably isn't a solute, not a solute, probably isn't an ion, and has a little bit more structure than a very small atom or a small molecule like water, is probably an inclusion, a chemical substance that can be present depending on the cell types. Glycogen is the stored form of glucose. Stored nutrients, lipid droplets, pigments, water-containing vacuoles, etc. Questions? You'll find that a lot of this chapter from here on out is a lot of terminology and just vocabulary. The mitochondria is the power plants that provide most of the cell's ATP through aerobic respiration. What does aerobic mean?
Contains oxygen.
Oxygen. I was going to start singing, let's get physical, but I'm not going to. But yes, so aerobic meaning it requires oxygen. Anaerobic meaning in the absence of oxygen. So mitochondria has two membranes. It has an inner membrane that folds inward, forming the cristae. So here's my outer membrane. Here's my inner membrane. That folds in and protrudes into the matrix. Inside here is the matrix. So we produce two different compartments. Mitochondria are amazing in that they have their own DNA and RNA and can reproduce themselves. They're the only organelle that we have that has outside of the nucleus where DNA is contained. So they have their own ribosomes, they have their own, which we'll talk about next. They have their own ribosomes, they have their own DNA, and they have their own RNA. As the cell's need for energy increases, mitochondria can increase their number of cristae, meaning these inner folds, or divide by fission to increase the number of mitochondria. Cells like muscle that require a lot of mitochondria because they're constantly breaking down ATP and constantly using it to allow for muscle contractions have a lot of mitochondria. Cardiac muscle has more mitochondria than skeletal muscle has. So the density of mitochondria in a cell reflects that cell's energy requirement. So ribosomes. Ribosomes are made of two subunits that are composed of both protein and rRNA or ribosomal RNA. These are sites for protein synthesis where the mRNA is read and translated into protein or a sequence of amino acids. Some ribosomes are free in the cytoplasm and will synthesize proteins that function in the cytosol. Others are found on the surface of the rough endoplasmic reticulum, and they synthesize proteins that are destined to be incorporated into cellular membranes or exported from the cell. The endoplasmic reticulum is a network within the cytoplasm. And it is an extensive system of interconnected tubules and parallel membranes that includes closed fluid-filled cavities called cisternae. This membrane of the endoplasmic reticulum is continuous with the nuclear envelope that surrounds the nucleus. And the enveloping, or the membrane of the endoplasmic reticulum, both the rough and the smooth, and the membrane of the nuclear envelope accounts for about 50% of the cell's membranes. There's two types of endoplasmic reticulum. There's rough ER that has little ribosomes embedded on its surface, giving it the appearance of being rough, and smooth ER, which does not have ribosomes on its surface. Proteins that are assembled on the rough ER, because ribosomes bind to the surface of the rough endoplasmic reticulum, will ultimately leave the cell or become part of the plasma membrane. So proteins that are assembled on the rough ER will move into the fluid-filled interior of the cisternae. Ribosomes make all of the proteins secreted from the cell. Proteins that will end up leaving the cell are produced by ribosomes and that then bind. So bear with me here. The protein binds to the ribosome. So let me back up. The mRNA binds to the ribosome. At that point, if the protein is destined to leave the cell or become part of the plasma membrane, it will get what's called a signal recognition particle attached to it. And that will actually halt translation of the protein until it's attached to the membrane of the endoplasmic reticulum. Once it's attached to the endoplasmic reticulum, the signal recognition particle will be removed. And it will be recycled. The protein that's being synthesized on the endoplasmic reticulum it'll finish being created, and then it'll move into the inside of the endoplasmic reticulum. So all mRNAs initially bind to ribosomes that are free in the cytoplasm. If the protein is destined to stay and function within the cell, it will be synthesized on ribosomes that don't attach to the endoplasmic reticulum. If the protein is destined to leave the cell, then it'll get a signal recognition particle attached to it. And then that will then bind to the endoplasmic reticulum. And that protein initially, before it's released from the cell or added to the cell membrane, will enter the rough endoplasmic reticulum set interior, or the lumen. Is this clear? The other function of the rough endoplasmic reticulum is that it's a membrane factor. So if the protein isn't going to leave the cell via exocytosis, it's going to become part of the plasma membrane, and that it'll be an integral protein or a phospholipid. The smooth endoplasmic reticulum are tubules that are arranged in a looping network with enzymes forming part of the membranes. The smooth ER metabolizes lipids and synthesizes cholesterol and synthesizes lipid components of lipoproteins. The smooth ER is also going to synthesize steroid-based hormones. Remember, the precursor for all steroid-based hormones is cholesterol. So it's going to create the cholesterol. The smooth ER is going to synthesize the cholesterol and then make the steroid-based hormones. It's also going to absorb and synthesize and transport fats in intestinal cells. In the liver, the smooth ER also detoxifies drugs, certain pesticides and carcinogens in the liver and kidneys. Much more in the liver than the kidneys. And it breaks down in other cells, such as the liver, is going to break down glycogen molecules. Remember, glycogen is stored glucose into free glucose so that it can then leave the cell. Questions? The sarcoplasmic reticulum is a specialized ER that functions in calcium storage that can be released during muscle contraction. The Zena can tell you from Biology 122, it's vital in the heart. It's important in the heart. It's also important in your skeletal muscle. So the Golgi complex, or the Golgi apparatus, is a stacked, flattened membranous sacs that are associated with swarms of tiny membranous vesicles. It's the principal director for cellular proteins. A lot of times we like to think of this as being the post office for the cell. Proteins that are going to leave the cell that are made in the cell, and then they're going to leave the cell. They enter the rough endoplasmic reticulum. And then when they're done being modified in the rough endoplasmic reticulum, they will then enter the cis phase of the Golgi apparatus, where the finishing touches on lipid and glycoprotein synthesis will be done, and any carbohydrates that need to be added will be added. And then these proteins are released by the pinching off of the cisterns of the Golgi apparatus. So they fuse to one side where you're going to get finishing touches on proteins and glycoproteins. Carbohydrates will be added if they need to. Splice some proteins if they need to. In other words, we'll cut them. We'll sort them, and then we'll pinch them off into vesicles from the trans face of the Golgi apparatus. From there, they can go in 3 different ways. The proteins are tagged, and they can either be sent to the plasma membrane, where they will be secreted via exocytosis, or they can be sent from the Golgi apparatus to the plasma membrane and become part of the plasma membrane. Or if there's something wrong with it, it can fuse with the lysosome and be destroyed. We don't want defective proteins out there. So if there's a problem with that protein, it will fuse with the lysosome and be destroyed. So three different things. Can either, once it leaves the Golgi apparatus, it can either become part of the plasma membrane. It can be brought to the plasma membrane and released via exocytosis. Or if there's something wrong with it, it can fuse with the lysosome and be destroyed. The lysosomes are safe places where digestion can proceed. That rises from the budding off. Hi. Sorry, I'm saying hi to my really old dog. She can't see. So safe place where digestion can proceed that arises from the budding off of the Golgi apparatus. So this is essentially the stomach or the recycling plant for the cell. It contains digestive enzymes, acid hydrolases, that digest proteins, nucleic acids, carbohydrates, phospholipids, and other substrates. The membranes contain these ATPase hydrogen ion pumps that are going to gather hydrogen ions from the surrounding cytosol and pump them into the lysosome. These are enzymes that have an optimal pH that's really low. And so we keep this recycling self-contained with these enzymes that are really great, things that breaking things down, nucleases, nucleic acids, proteases, proteins, glycosidases, carbohydrates, lipases, lipids, phosphates, phosphatases, things that have phosphates attached to them get broken down. All of these are named for what they do. Phospholipases break down phospholipids. So we pump hydrogen ions into this organelle. And what we end up with is a very acidic environment with enzymes that have optimal pHs that are very acidic. Therefore, they're really great at this lower pH, and they break things down. And the result is that you can safely break down things and keep these enzymes contained, but they also don't work well at the higher pH of the cell, the 7.2. So the lysosome is the cell's demolition crew. It digests particles that are taken in by phagocytes, such as ingested bacteria, viruses, and toxins. Autophagy, so being able to degrade and recycle worn out and non-functional organelles. Organelles get old and they need to be recycled. The lysosome can also perform metabolic functions such as glycogen breakdown and glucose release, in addition to the smooth endoplasmic reticulum, which also helps with that. Also, you can break down non-useful cells. So the lysosomal membrane can become unstable when the cell is deprived of oxygen or excessive amounts of vitamin A are present. And the result is that the lysosome ruptures, resulting in autolysis of the cell. I think this was one of Justin's questions in a previous lecture. When do you actually want breakdown of cells? One of them is to eliminate the webbing in between your fingers and toes. The other one is to break down the uterine lining during menstruation. Another reason that we'll see a lot of in the next chapter is there's cells in the bone that are specific for breaking down bone, osteoclasts, that break down bone by releasing calcium into the blood. But in order to break down that bone, they have to bring the bone in and degrade it, and they degrade it using the lysosomes. So osteoclasts, remember that class meaning breakdown, osteo meaning bone. So osteoclasts have an exceptional amount of lysosomes in them because they're good at breaking down bone. Tay-Sachs disease, genetic disorder, where lysosomes lack the enzyme needed to break down glycolipids that are abundant in the nerve cell membranes. As far as I know, there's no cure for it. It's very common among more Jewish populations. I'm reading a book about genetics right now called You Have Your Mother's Laugh. It's a recent book put out by a guy who's explaining genetics. So if you like those kind of books, this is another one. I said that to my biology 122 students how much I recommend the book about the immune system. And if you're interested, it's a great book too. But this one that I'm listening to right now is all about genetics. And this is one of the diseases he brings up. So Tay-Sachs is a genetic deficiency of an enzyme in the lysosomes that breaks down glycolipids that are abundant in nerve cell membranes. Because the enzyme is deficient, the lysosomes swell with undigested lipids, which ultimately will interfere with normal nerve function. And so the disease, from the time that it is diagnosed within 18 months, will progress through mental retardation, seizures, blindness, and ultimately death. I'm guessing this is one of those diseases, and I'll bet you if you looked it up right now, it would show it that they're working for gene therapy with it, or looking at investigations into genetic therapy for it. Peroxisomes, similar to lysosomes, but they're a little different in that they use oxidases that use oxygen to oxidize things. Remember, we talked about oxidation reduction reactions. So they use oxygen to oxidize things. They can also neutralize free radicals and convert them to hydrogen peroxide, which is then broken down into water. So free radicals and hydrogen peroxide are normal byproducts of cell metabolism, but are toxic when allowed to accumulate. They self-replicate by pinching off from pre-existing peroxisomes. So the easiest way to remember these is they are going to produce hydrogen peroxide. They break down lipids specifically. So they're especially numerous in the liver and in kidney cells that are really active in detoxification. Just like your smoothie are. This is another one. Peroxisomes are another one that's very numerous in the liver and kidney because they're good at detoxifying things. I think we should take 5 minutes here. I don't want to take more than 5 because I really want to get through chapter 3. But let's take 5 minutes here and come back to right here. Is that all right with you?
Yes.
Ruby, do you need to go outside, baby? Lauren, I just thought about what I said a second ago about vaccines. I need to take back some of that stuff. I sometimes have a really lousy filter on my mouth. And
Okay.
well, I think sometimes, especially with vaccines killing children, like I really don't know what it's like to be a parent. I know I've dealt with really sick kids sometimes, and I can tell you it, having my limited experience with really sick kids, it's kind of scary.
I'm pro-vaccine, so like
Yeah.
that doesn't bother me. But like my question was like, what would cause that allergic reaction that people get? Like, is it the chemicals like inside the vaccine? Like, you know how there's like preservatives in the vaccine vial to like preserve the
Well,
vaccine.
I think there are a little bit, but more than anything, your body's going to respond to something that's foreign.
Okay.
And that's what an allergy is when your body thinks that something is wrong with it and react. And like a normal allergy response, your body senses that something is foreign and dangerous when it shouldn't be, like pollen, like cat hair.
Mhm.
And
But
often in vaccines, Lauren, it's like proteins that you are allergic to. It's very, very rarely like the individual components. It's more so the more organic compounds that your body has that reaction to. Like it's really common to have like an egg allergy, so you have to have an egg-free vaccine. So that's a big one.
But I still think, Justin, I still think that on some level, all immunizations work like an allergic response. But it's not so much an allergic response as your body recognizes that it could be harmful. And that's why you develop a response to it.
And is that how we gain the like? I
Yes.
wasn't sure if my question was is considered microbiology, because I guess that can go into micro territory. But from that reaction and from the dead virus that's introduced, is that how our body knows that when we experience this, whatever it is in our body, they know to fight it? Is that where that reaction
That's where it comes from? Yep, that's exactly where it comes from. And that's why secondary allergic responses can be really bad also, is because your reaction is so much worse the second time around.
Huh.
The reason I thought about what I said, as soon as I got off, I walked in the other room and I remembered a student telling me about a professor at one of the colleges in Maryland who was taking a week off because her baby daughter had just died of a vaccination response.
Oh no.
And so I know it happens. And I don't know, I know like my little nephew who almost died. He was in the hospital for a month after getting a vaccination when he was 3 months old. And I was thinking about it a second ago. I have a little niece who has Williams syndrome. And I didn't know this until recently, but she can't get vaccinated because little kids with Williams syndrome tend to do very poorly with vaccinations. And so I think I think a lot of reason which people should get vaccinated. One more thing for my soapbox is to protect those who cannot be vaccinated. Because what it essentially says is that the infection stops with you. Does that make sense?
Yeah, that makes sense. My clinic, we routinely we give out both COVID vaccines, and then like for our new patients, like if we find that they're not if they don't have like the hepatitis antibodies, we also give them the Hep B like vaccine as well, because it's like we deal with people's blood outside
Yeah.
of their body, and
Right.
so like, oh yeah, you should get this because it'll prevent you from catching Hep B, because even though we try to sanitize and we isolate the patients with Hep B, there
Right.
is still that chance,
Right. Okay. I understand that. Okay. So coming back, the cytoskeleton works like your skeleton does in a lot of ways. It acts as the cell's bones, muscles, and ligaments to support cellular structures and provide machinery that's going to generate cellular movements. So it's what allows you to create those pseudopodia. Actually, did we talk about pseudopodia? I don't think we have. I'm getting myself confused with another class. But we talked about those cell adhesion molecules that work as arms to allow cells to haul themselves over other cells. Well, part of what's needed there are changes in the cytoskeleton. Because think about it, if you're going to move out like this to grab hold of something with your cell adhesion molecules and then pull yourself forward, you're going to have some major changes in the skeleton. So there's three basic types of rods. There's microtubules, microfilaments, or actin filaments, and intermediate filaments. Microtubules are the rods with the largest diameter. They're hollow tubes that are made of spherical tubulin subunits. And most radiate from centrosomes. They determine the overall shape of the cell, distribution of organelles, and chromosome movement during cell division. They're continually pulled and moved along by motor proteins that move along the microtubule monorail. So these work like streets and paths where motor proteins use them to move a structure from one place to another. And rather than just being static, non-changing roads, these are changeable roads that continually assemble and disassemble. Microfilaments are the rods with the smallest diameter, and they're made of actin. They're involved in cell motility, changes in cell shape, and strengthen the cell's surface. So these are what are going to make those little arms that you're going to use to pull yourself over something if you were a cell. They form the terminal membrane, the membrane skeleton on the cytoplasmic side of the plasmic membrane. Actin filaments, these microfilaments, actin filaments interact with myosin to to form cleavage furrows during cytokinesis, during cell division. They attach to cell adhesion molecules to produce the crawling movements, develop these little pseudopodia. Brought it up before, and I shouldn't have because we weren't there yet. But pseudo meaning looks like but isn't. Podia meaning feet, so these are false feet. That's a picture of an amoeba, but you have cells that are very similar. A lot of your white blood cells have the capability of forming pseudopodia. So they attach and produce crawling motions and allow for endocytosis and exocytosis. Just like the microtubules, they constantly break down and reform and interact with myosin to produce the contractions observed in muscle cells. Intermediate filaments are rods of intermediate diameter. Woven ropes with high tensile strength. They attach to the desmosomes. Remember, those were a cell-to-cell junction, and act as guy wires to resist pulling forces exerted on cells. You remember we talked about desmosomes and how they were involved in attaching cells to cells and helping resist separation of those cells. Well, part of their Velcro component of the desmosome are these intermediate filaments. They're called neurofilaments in nerve cells and keratin filaments in epithelial cells. So just an overview. Microfilaments are the smallest. They're strands made of spherical actin. Intermediate filaments are tough, insoluble protein fibers. These, you find them on desmosomes or near desmosomes. They're constructed of a four tetramer fibro corded up rope. And microtubules, which are of the largest diameter, which are made of spherical subunits called tubulin. Questions? So organelles, centrosomes and centrioles. Microtubules, remember, they're generated by centrosomes. And centrosomes are granular matrices in the cytoplasm near the nucleus that contain a pair of barrel-shaped centrioles. So centrioles. They're always found at right angles to one another, the centrioles. So the centrioles are what are actually going to create the microtubules. And the centrioles are contained in the centrosomes. So let me say this one more time, and I'm going to say it a little differently. Centrosomes contain the centrioles that generate the microtubules.
That generates what? I'm sorry.
Okay, yeah, let me just say it again. So centrosomes contain centrioles that generate, where's my mouse? There it is. There it is. Microtubules.
Thank you.
You're welcome. So centrioles are composed of pinwheels of 9 triplets with a hollow inner tube. 9 triplets. So 1, 2, 3, 1, 2, 3, 1, 2, 3, 9 of them all together. And they generate the microtubules and organize the mitotic spindle that's made of microtubules. They also form the basal bodies of flagella and cilia. Cilia are whip-like motile cellular extensions occurring in large numbers on the cellular surfaces. These have a really great immune function. In combination with mucus, they're amazing. And they move substances in one direction across the cell surfaces. For example, you have cilia that line the respiratory tract. You have cilia that line the fallopian tubes. When the centriole projections are significantly longer, or cilia are significantly longer, they're called flagella. Sperm are the only human cells with flagella. So cilia will propel themselves, will propel other things again along the cell surface, whereas flagellum propels the cells themselves. So cilia work like little hair-like structures. Flagellum work like little swimmers. The pattern of microtubules in the cilium and flagellum align themselves so that you have 9 pairs of doublets encircling one central pair. Bear with me here, because I don't like the way that's worded now that I'm reading it. Here, cilia, microtubules, 3 sets of triplets. Excuse me, 9 sets of triplets. Gotta read. 9 sets of triplets, or count. 9 sets of triplets. On the other hand, as we move down the flagellum, initially at the very beginning of the flagellum, it's 9 sets of triplets. However, as you move down, those 9 sets of triplets will actually become 9 sets of doublets with the 1 in the center. So I guess I just don't like the way I've put this in here. Because ultimately the flagellum is the one that's going to have 9 pairs doublets with encircling 1 central pair. The cilium come with 9 triplets. Extending from the microtubule doublets are arms composed of dynin, motor protein, that grips the doublets, and starts to crawl along their length. The length bends the doublets, which then collectively result in the cilium bending. So you have these little, if you zoom in here a little bit, you can see the little red here, working like little motor proteins, attaching onto one and climbing down the other one. And the result is that you bend them. So pairs of dynon arms attach onto one and bend and climb with the other one and ultimately bend it. You know what? I'm going to do something really quick. I'm struggling with the way I'm saying that. So I'm going to look on something else really quick. Because I thought I had a good video of this. Bear with me. And I'll post it to this site later if it's here. Maybe I didn't put it on there. All right. Bear with me just for a second here. See what this one is.
Good.
Okay, that's terrible because I want it to have words. Just a sec.
So I've got my essay written. And the cytoskeleton of flagella and modal cilia is composed of microtubules. These microtubules are arranged.
You know, I think the structure changes. So it goes from being 3 triplets to becoming, assuming 9 triplets to becoming 9 doublets with an inner circle.
Around 2 singlets or 2 single microtubules surrounded by a ring of 9 doublets. These are attached to each other by proteins known as nexins so that they don't move relative to each other. And the protein 9-nein uses its globular head to pull one neighboring microtubules to allow movement. There are also radial spokes and other proteins involved. And flagella and cilia are attached to a basal body, which has a similar structure, but which lacks these singlets in the center.
Okay, these are terrible because I know I've seen it better, and so I'm not going to try to find one right now. But I'll work to find what I know I have.
Let's talk about microtubules in more detail. So first we'll discuss the.
I know I have better than what I'm showing. But just realize that one anchors on one of the doublets and it pulls on the other doublet and it crawls along the surface, and the result is that you bend it. So the crawling bends the doublets, which then collectively results in the cilium bending. This bending alternates rhythmically between a propulsive power stroke and a recovery stroke. The cilia push down, so they bend down, one little doublet crawling down another doublet, forces it down, and then it flips back up. So this alternating rhythmic action creates this pushing motion in a single direction that's repeated 10 to 20 times per second. The bending of one cilia is followed by the bending of the next and the next, resulting in a traveling wave along the cell surface. The nucleus is the gene-containing control center. It contains the instructions needed to build almost all the cell's body's proteins, dictates the kinds and amounts of proteins needed to be synthesized at one time in response to signals acting on the cell. Cells can be uninucleate, having one nucleus, multinucleates, like skeletal muscle and osteoclasts and liver cells, or anucleate, like mature red blood cells. Without a nucleus, red blood cells can't regenerate their mRNA, they can't replace their proteins, and so they gradually deteriorate because they can't reproduce and therefore must be replenished. Recognizable structures, you have the nuclear envelope, the nucleoli, which contains the ribosomal RNA, is involved in making the ribosomes, and chromatin. There's several distinct compartments that are rich in a subset of proteins that are always in a constant dynamic flux. The double membrane envelope is continuous with that membrane of the endoplasmic reticulum. It has an outer nuclear membrane and an inner nuclear membrane. The inner nuclear membrane is lined by the nuclear lamina, which is similar in a lot of ways to the cytoskeleton next to the plasma membrane, in that it's a scaffolding network of proteins that help maintain the shape of the nucleus and organize the DNA in the nucleus. Just like the plasma membrane, this envelope is selectively permeable. But the passage of substances much more freer here between the inside and the outside of the nucleus than that of the plasma membrane. The two layers of the envelope, because it's actually a continuous membrane, meaning that it folds on itself. The plasma membrane doesn't do this, but this membrane is going to fold on itself. And when it folds on itself, it's going to create pores, or it's going to be where it folds is going to be the location of pores. The proteins that line each nuclear pore create a channel, regulating large particles in and out of the nucleus. Protein molecules are imported from the cytoplasm, and RNA molecules are exported from the nucleus via nuclear pores. So this is the mRNA, before it's made into protein, has to leave the nucleus. It's going to leave via these pores. The ribosomal subunits that are created in the nucleus within the nucleoli have to leave this nucleus before they can take the mRNA and produce the protein from reading the mRNA. Nucleoli, typically we have one or two. They contain the DNA with the genetic material that's necessary for synthesizing ribosomal RNA or rRNA that will be combined with proteins to make ribosomal subunits. Most ribosomal subunits will exit through the nuclear pores and enter the cytoplasm. We're going over, we're just covering a lot of material. We're going over the basics of it. You're going to learn enough about cell biology so that you can apply it later. So chromatin and nucleosomes. Chromatin is a system of bumpy threads composed of 30% DNA, 60% globular histones, and 10% RNA chains. The nucleosomes are an octet of histones that have the DNA double helix wrapped around them twice. So chromatin is a linked structure of nucleosomes. Histones play an important role in regulating gene expression. They are key to turning genes off and turning them on. Every one of your cells contains all of your genetic material that you were born with, outside of your red blood cells, which don't have a nucleus. All of your other cells contain all of the genetic material you were ever born with. However, certain genes are turned off. These are what play a role in turning things off and turning them on and regulating what protein should be expressed in what cells. Extended chromatin, or what we call euchromatin, euchromatin, is active and not easily visualized. Condensed chromatin or heterochromatin is inactive and stains dark. And I'm going to stop just for a second. So euchromatin is extended and open chromatin. That contains acetylated histones. It's actively expressed DNA. So it's DNA that's being actively made into proteins or replicated. Actively made into genes, made into proteins or replicated. DNA that is actively made into proteins or replicated. Heterochromatin is condensed closed off chromatin that contains methylated histones and this DNA is not actively made into proteins or replicated. So euchromatin is extended open chromatin that contains acetylated histones. It's DNA that is actively made into proteins or replicated. Heterochromatin is condensed closed off chromatin that contains methylated histones. This is DNA that is not actively made into proteins or replicated. Okay, I'm gonna go back. I know you guys are writing that down and I really want to get through this chapter. So I'll come back to it at the end. I won't clear the screen. I'll come back to it at the end and you can write down whatever you've missed there. So when a cell is preparing to divide, the chromatin condenses into chromosomes. So that's what we actually, when we look at linear chromosomes, what we're looking at is the condensed chromatin. And this condensation of the chromosomes prevents entanglement and breakage during cell division. Cells synthesize a variety of proteins that determine the chemical and physical natures of cells. A gene is a segment of DNA carrying the instructions for creating a polypeptide chain of a protein or a variety of RNA structures. The information in the DNA is carried in a consecutive strand of triplets. Each sequence of 3 bases code for the order for each of the amino acids within a protein. Genes contain exons, which are the amino acid specifying sequences, and introns, which are non-coding and often repetitive sequences that have been shown to actually have many functions. Initially, we just wondered what their purpose was. Were they junk DNA? Well, coming to find out, they're actually involved in regulating genetic expression. And also we have multiple genes that can be made into, one gene can be made into multiple proteins by cutting out different parts of the gene, the mRNA, specifying different parts of the gene to be considered an intron can allow one gene to code for multiple different types of amino acid sequences. If you look at our genome compared to, oh, I don't know, a yeast genome, we don't have a lot of genes in comparison to some other animals, Drosophila melanogaster, the fruit fly. We don't have a lot of genes. But what we have is that genes are alternatively spliced and rearranged so that one gene can make multiple proteins that may have very, very different functions. All RNA is formed from the nuclear DNA. RNA can carry out different processes in the protein or polypeptide. mRNA, messenger RNA, are sequences where each of the three bases, a codon, along the RNA strand are going to code for one amino acid within each polypeptide chain. So this is the intermediate that we take from DNA, make it into mRNA, and then we use that mRNA, we read it, it's read to make the protein amino acid sequence. Transfer RNAs are structures that actually are involved in this reading process with the ribosomes, where they will actually bind to amino acids and read the mRNA sequences and bring in the correct amino acids for each correct codon. Ribosomal RNA are part of the ribosomes. So the mRNA is your message. Transfer RNA brings in the right amino acids for each part of the protein, each amino acid of the protein. The ribosomal RNA is involved with the ribosome, which is involved in catalyzing the bonds between each amino acid. So how are genes created? 2 steps: transcription and translation. Transcription is where the DNA is transcribed into an mRNA in the nucleus. Translation is where the information carried in the mRNA leaves the nucleus, goes out into the cytoplasm, and is decoded or translated and used to assemble the polypeptide. Honestly, I'll tell you that what we cover in here in this one chapter, or in the last 2 chapters, or even the last 3 chapters, is the majority of what I teach in Biology 101, which is a general biology course for most schools. So we're covering a lot. We're just going over the top of a lot of material here. So this is a process of transcription to translation. So DNA, based on the coding of the bases and base pairing, you make an mRNA transcript. The mRNA transcript is then read via the tRNAs, and the bonds between the individual amino acids, where each of the codons, 3 bases, read for, are catalyzed and synthesized via the ribosomes. Questions? cytosolic protein degradation. So we like to have proteins, but they can outlive their usefulness and then they need to be destroyed. These proteins can be destroyed via the lysosomes. Misfolded, damaged, or unneeded proteins in the cytosol can be marked with ubiquitins for proteolysis. You guys, what does proteolysis mean? What does lysis mean?
To eat or decrease it, break down.
To break down. So lysis means to break down. So proteo meaning break down. So proteolysis is break down proteins. Ubiquitins are these tags that are everywhere. That's where they got their name. You know, ubiquitous means that something that's everywhere all the time. That's what these are. So the misfolded, damaged, or unneeded proteins are marked with these ubiquitin tags that are then marked for degradation with this ubiquitin tag. And then they undergo proteolysis in an enzyme called the proteasomes that degrades small or large proteins. Small proteins can maybe just be degraded by proteases. Larger proteins must be degraded by proteasomes. Extracellular materials. So now we're talking specifically about materials outside of the cell. Any substances, extracellular materials by definition, are any substances that contribute to the body mass that are outside of the cell. Body fluids that function in transport and dissolving, such as interstitial fluid, blood plasma, and cerebrospinal fluid. Cellular secretions, such as intestinal and gastric juices that function in digestion. Saliva, mucus, and serous fluids. Extracellular matrix, composed of proteins and polysaccharides that can be hard or soft and function as cell glue or medium for cell signaling. Something to always keep in mind, embryonic stem cells. In various regions of an embryo, so bear with me, stem cells are found in various regions of an embryo and even in human adults that can be exposed to different chemical messengers that then will channel them into specific pathways of differentiation. Another word for that helps with understanding differentiation is specialization. You have a doctor who then can specialize into a specific type of doctor, or a nurse that can specialize into a specific type of nurse. Chemical messengers can be slight differences in oxygen and carbon dioxide concentrations. These cells then release chemicals that will influence the differentiation of neighboring cells through altering the gene expression in different cells. So the major player behind cell differentiation is going back to the fact that each one of your cells has your full genome, all of your genetic material. But it will become a certain type of cell and differentiate into a certain type of cell by the turning on and off of specific genes. So cell differentiation is this development of specific and distinct features in cells, such as increased amounts of organelles or myosin and actin. So cells, depending on what are they, the first thing that happens is you go from being a ball of cells to being an organized ball of cells that has different layers to it. If you want to take the inner cell mass out, you can remove the inner cell mass, culture it up, and allow it to become muscle cells, blood cells, neurons, intestinal cells, pancreatic islet cells, liver cells, depending on what chemical signals you treat it with. Any questions? Apoptosis. So Max was the one who asked about this, and he's not here tonight. But process of uncontrolled cellular suicide that eliminates stressed, unneeded, aged, or excessive cells. So damaged macromolecules or an extracellular signal will activate a series of caspases that then destroy the DNA and cytoskeleton. The cell will shrink, detach from other cells, round up, and start to bleb. Meaning blebbing, just kind of little pieces falling off. Dying cells will release chemicals in apoptotic bodies that attract macrophages. Phagocytosis, remember that cell eating, macro meaning big cell that eats. So macrophages will be attracted, and then the dead dying cells will be phagocytized. Cancer cells are really good at avoiding apoptosis. Remember, they can change their glycocalyx, but then we have cells of the immune system that will look at a cell and think, okay, is there something wrong with the cell? Obviously, there's something wrong, and they kill it. Whether they realize what's wrong or not, they still destroy it. However, heart muscle during heart attacks and nerve cells undergo strokes. During strokes undergo apoptosis. Let me say that again, sorry. However, heart muscle during heart attacks and nerve cells during strokes undergo apoptosis with an oxygen deprivation signal. All of a sudden, there's no oxygen coming in. It's deprived, it gets hypoxic, and it stimulates apoptosis. I want to say that almost all cells, including the cells between your fingers that undergo apoptosis, it starts with an oxygen deprivation signal. Local changes that can change the rates of cell division. Hyperplasia, accelerated growth. With anemia, bone marrow undergoes hyperplasia so that red blood cells are produced at a faster rate. Remember, we talked about how that yellow bone marrow that's in between in your bones can start to become red bone marrow. That's hyperplasia. Depending when you need it. Atrophy is a decrease in the small size of an organ or body tissue that can result from a loss of normal stimulation. Muscles that lose their nerve supply will atrophy and waste away. A lack of exercise can lead to thinned and brittle bones. Questions? Cell aging. So the wear and tear theory attributes cell aging to chemical insults and the formation of free radicals that accumulate over time. The result is that you can get mistakes in DNA replication. Most free radicals are produced in the mitochondria and energy production, so they're formed from normal everyday reactions. You're gonna form them. The point is, are you able to neutralize them? So most free radicals are produced in the mitochondria and are diminished. Energy production will be diminished in radical damaged mitochondria. Also, temporary lacks of oxygen in blood vessels causes them to clog with fatty materials, leading to accelerated rates of cell death. Let me back up just for a second. So mitochondria is your powerhouse, and it's the one that produces the most amount of free radicals. When you're not able to neutralize them, then your organelle essentially gets overrun with free radicals. And you can see how that's going to hurt the energy production of that organelle. Last slide. Telomeres, another hypothesis of cell aging. Telomeres are on the ends of linear chromosomes. The longer the length, the more stable that chromosome is. We often look at them as being a telomere clock because every time a DNA is replicated, 50 to 100 of the N nucleotides are lost and telomeres become shorter. When the telomeres reach a certain minimal length, a stop signal is given and the chromosome is no longer able to divide. We have immortalized cell lines that will replicate over and over and over again and over again and over it again and over again, so for forever. Their telomeres don't shorten. If you look, and I think I actually took it out, but I had it in here earlier, but I needed to make this a short PowerPoint. If you've ever heard of progeria, someone nod at me and say they understand what I'm talking about. Progeria?
Yes.
Okay. What they have noticed, so those are children that age really, really early. You've seen them look like little tiny people that look like little grandparents. They have linked telomere length to progeria. Questions?
Isn't it where they their cells age really quickly? That's why they look like.
That's just one possibility. Whether it's the reason why or it's a symptom of what's already going on, they don't know, but they know that it's linked. Does that make sense?
Yes.
Yeah, I did a little bit. When I saw that, I think it might be in your book. I'm not sure. But I did a little research into it, and it's linked, and whether or not it's the cause or whether it's other things that just result in this, who knows? But we do know that immortalized cell lines that will continue to replicate and replicate and replicate for forever don't change in their telomere lengths on their chromosomes. Guys, have any questions for me about chapter 3? It's a lot of material, but it's also not as hard as some of it was to comprehend. It's just a lot of it. But I have faith in you with the exam, because the person who I saw who's already taken it did really well on it. I want to start into chapter 7 and spend the last 10 minutes on chapter 7, unless you have questions for me. I think you will start seeing that our chapters are going to start going a lot faster. I think the osmosis was the more difficult part of, and the slower part of this chapter, which I kind of expected it to be, because it usually tends to be the harder part for a lot of classes. But I think you'll see that we will start moving through material a lot faster. So chapter 7, bone tissue. Some of this, at least the beginning or part of the beginning, is going to be a review of lab from the other night. So bone tissue. The skeletal system consists of bones, cartilage, and ligaments that are tightly joined together. The cartilage is the forerunner of most bones in the embryo skeleton and bone development in childhood, and also covers the joint surfaces in mature skeletons. The ligaments hold bones together at the joints. One thing that we should mention right now, and then it won't come up again later, or it might still come up, but we'll go over it again when we get there. Tendons connect muscles, or oh shoot. Yeah, tendons connect muscles to bones. Ligaments connect bones to bones. That's why tendons, that's why tendons aren't considered part of the skeletal system, is because they're attaching muscles to bones. Sometimes they attach muscles to muscles, but mostly muscles to bones. So tendons, bones, cartilage, ligaments. Ligaments hold bones together at the joints. Tendons attach muscles to bones. Which of the following tissues could be found on or within a bone, such as the humerus?
Is it all of them?
It is all of them. Good job. It is all of them. I was gonna say go and think back about what we did in lab, but it's all of them. So the skeleton is a composition of multiple tissues: nervous tissues, yellow bone marrow, blood, compact bone, dense regular connective tissue, red bone marrow, cancellous or spongy bone. Also, bones continually remodel themselves and interact physiologically with all other organ systems. Therefore, each individual bone can be considered one organ because it's composed of different tissues working together. Different functions of the skeleton. Skeleton supports the body, teeth, and viscera or organs. It supports soft tissue and provides attachment points for tendons of skeletal muscle. It protects and encloses internal organs such as the brain, the spinal cord, the lungs, the heart, the pelvic viscera, and the bone marrow. So support, protection, movements, including those involving the limbs, breathing, and other movements where muscles act on bones. Also, one thing that we'll spend a lot of time talking about is that bones are really important in balancing electrolytes because they store calcium and phosphate or inorganic phosphate and allow their release into the tissue fluids. So support, protection, movement, balance electrolytes, acid-base balance through buffering the blood against excessive pH changes by absorbing or releasing alkaline phosphate and carbonate salts. And blood formation in the red bone marrow where blood and the immune cells are produced. So protection, support, movement, electrolyte balance, acid-base balance, and blood formation. Hematopoietic. I'm going to introduce some more terms here. Hemato means blood. It's right up there with the word hemo, which also means blood. Poetic means producing. I don't think we've talked about these before. So hematopoietic cells are blood producing cells. So this is hematopoietic tissue, is a tissue in which new blood cells are going to be formed. Which of the following functions of the skeletal system would be considered a metabolic function?
D.
Say it one more time.
Is it D?
D. What would be my answer here? I'm going to say no. And the reason is I think it's mineral storage. But you know what? I would not ask this question on an exam. I think I would honestly say both of these, but I think C is more correct because that's the electrolytes and metabolism. Does that make sense? Maybe.
Yeah.
So types of bones, stay with me for just 3 more minutes here. We have long bones, which have a greater length and width and consist of a shaft and a variable number of ends. We have short bones that are somewhat cube-shaped and nearly equal in length and width, like our carpal or our tarsal bones. We have flat bones, like the bones of our cranium and our sternum, that are generally thin and afford considerable protection. We have irregular bones, like our vertebrae, or our maybe not our sacrum, our I guess you could maybe say your os coxa or your pelvic bone, that have complex shapes and cannot be grouped into one of any of the previous bones, long, short, or flat. Two different types of bone is found in long bones. It's also found in other bones. We also have compact and spongy bone in all of the bones. It's easier to describe in the long bones, but recognize that it's found in all of the bones. Compact or cortical bone is the outer shell of dense white osseous tissue. It encloses the marrow or the medullary cavity that contains the bone marrow. Spongy or cancellous bone, which is in the central space at the ends of the long bone and just inside the compact bone in the diaphysis or the shaft. And you guys, I think this is a really great place to stop. Let's stop here. We will pick up, tomorrow's lab is going to be fairly short. And so I want to do a little bit more lecture tomorrow night, but we'll pick up right here. Do you have any questions for me?
As tomorrow, you mean Wednesday, correct?
That's what I meant to say. It's just not what came
No problem.
out. Yes.
Professor Gessel,
Yeah.
I just wanted to point out that there's 2 instances of the Chapter 7 quiz.
Are there? Oh, wow. Okay. Just
One is closing tonight, and so that's why I was bringing it up.
you know what? It sometimes when I extend out a due date, it gives you 2 different due dates.
Right, and I know that sometimes, like if the due date comes up, then it'll automatically give you the grade. So I just wanted to bring it up. I didn't know if it needed to be deleted.
If I can't delete it. That's the
Oh.
thing. I can't delete it. Once I extend it, it's there. But if you run into a problem with it, let me know.
Okay, thank you.
Of course,
Have a good night.
you too.
I did have a question for you. I reached out over text message and I sent a video. It was in regards to the chapter 7 quiz.
When did you, when did you do this?
I believe it was Sunday, Sunday or
Right.
Saturday. No, it was not Sunday. It was, hold on, Saturday.
All right, let me see if I got something. Um, what did you name it?
It was a text message. It wasn't an
Oh,
email.
was it? It was a text message. Darn it. I'm like, you said that. I thought I'd responded to every, oh, yep, I see it now. All right, what was going on with it?
This isn't giving any answers or anything. There was one of the last questions that I had gotten, it was a dragon